everybody. My name is Dr. Raymond Wong. I'm a clinical biochemical geneticist at Rady Children's Health in Orange, California. I am a principal investigator in a number of gene therapy clinical trials, primarily with adeno-associated viruses, and also run a translational research laboratory that assesses the use of somatic genome editing for various lysosomal storage diseases. So I'm super excited to bring to you today's community quick take in a topic that is very relevant to me. It is very exciting, generates a lot of hope, but also a lot of questions, which is somatic genome editing in vivo. So this approach really has, I believe, the potential to transform the lives of people living with genetic conditions. But just as with any other groundbreaking technology, it's very important to understand what opportunities this technology can unlock, but also its limitations as well. Today, we're joined by Dr. Sonia Vallabh, who not only brings deep scientific expertise, but also a very personal connection to this work. Her journey reflects the critical role that patients and advocates play in driving research forward, shaping research priorities, and ensuring that innovation really stays focused on real-world relevant impact. So let's get started and welcome.
Thank you so much.
Can you tell us a little bit about yourself and your journey, if you will?
Sure, absolutely. So today I run a research lab at the Broad Institute in Cambridge, along with my husband, Eric, and we are super mission focused on developing a therapy for prion disease in our lifetimes. Prion disease is a little bit famous. People might be familiar with it, but it's a rapid neurodegenerative disease. Most patients die within 6 months of their first symptom, and there's nothing we can do about it. But the kind of like unusual backstory is that I actually trained in a different field. I trained as a lawyer. My husband trained as a transportation engineer. And then shortly after we were married, this was happening in 2010, that my mom, who was 51 at the time, became really suddenly ill and deteriorated in front of our eyes at a pace that was unbelievable, and then died. And she died without a diagnosis, but it was only, afterwards from her autopsy that we learned that what we had seen was genetic prion disease, which we knew nothing about at the time. And not all prion disease is genetic, so that came as a total shock. But this was sort of our wake-up call that I was at 50-50 risk for the mutation that had caused her disease. So I got predictive genetic testing. And, for me, from the second that I understood the choice in front of us, that was the right choice, and ended up testing positive for her mutation. So this catalyzed a big change for us. And we ended up leaving our old careers behind to retrain in biomedicine, kind of from the ground up, constantly kind of reevaluating, like, how can we learn some of the tricks and tools that we need, but how can we keep sort of pointing ourselves and reorienting ourselves in the direction of a therapy for prion disease in our lifetimes? So we've been really, really lucky. We found our intellectual scientific home here at the Broad, and we've been here for many years now. We have a lab that I feel so lucky to lead and be a part of, that is 17 people, and we just throw ourselves at this every day.
Wow, that's a very personal connection. So tell me a little bit then about how potentially genome editing, well, first, can you tell the audience what genome editing is? maybe how that differs from gene replacement therapy or ex vivo gene editing, and then
Sure.
maybe we can look at how that plays into potential treatment for this condition.
Absolutely. So when I think about sort of our quest in context, I think we've been exceptionally lucky to be coming at this now. We were transitioning to science in the years 2012, 2013, 2014, just when sort of genetically targeted modalities were becoming realistic, in particular for the brain. We're a brain disease, we're a whole brain disease, and we have to reach as many neurons as possible. And so this was becoming, we were starting to see oligonucleotide therapies that were working for the brain. And even then, as we sort of leaned into the progress there, um, gene therapy remained to me somewhat hypothetical for our indication because reaching the brain is just so hard. And that's where it's really been the past few years where I feel like we've seen um, like really relevant advances. This has been an interesting ongoing conversation with my community because there are other areas where gene therapy has sort of been able to come online and get to patients sooner. And I think, some of the early test cases are editing cells outside the body in order to put them back into the body, right? And there you circumvent the delivery problem entirely, although there are a lot of other challenges, taking cells out, putting them back in. Um,
Mm-hmm.
and I think there are also organs that have been relatively easier to reach. The liver has been relatively easy to reach, the eye. So, you know, I think balancing excitement about those advances with the sort of like devil is in the details has been one of the tasks of how we message with our community. Um, I think that it's really in the past 2 years that I've started to say to our community, it's not going to be overnight, but I think there's a shot. I think delivery systems are being explored and invented now that have a shot at reaching enough of the right kind of cell in the right place because we don't have the option to take neurons out. We got to get to them and we got to get to as many
Right.
of them as possible. Um, in terms of what we do once we get there, we're lucky to have really simple genetics, molecular mechanism. Prion disease is caused by one protein. That's derives from a single RNA, a single gene, and we know it is gain of function. That protein goes rogue and it recruits other copies of itself. So, for a long time, and we were just lucky to have inherited a lot of sort of basic biology insight. For a long time, it's been known that every stage of that process depends on how much normal prion protein there is in the brain. And that is the fuel to the fire. And if we can dial it down, we are going to be protective. It's just a question of how much can we dial it down, how comprehensively can we do this across the brain, and how early can we get to people. So I think that defines the task in a way that still leaves a lot of options open. And when I think about gene therapies, now we're not trying to like replace a gene. We need to like get rid of
Sure.
a thing. But in
Right.
principle, we can do that through base editing. And this is a thing that we have explored is creating an edit in the prion protein coding sequence that creates a stop codon so that the whole protein
Mm-hmm.
does not get made. And we have like a nice ongoing collaboration with David Liu where we've shown like, yeah, that works. And then a different kind of editing that we've been exploring recently is sort of epigenetic editing to put marks on the gene that sort of shut it down and keep anything from being made from it. So that is a system called CHARM that we've been working on with Jonathan Weissman. On both sides, I think we can see the level of protein in the brain, coming down, and we can see that that is dose dependently protective. And um, really the key for both strategies, the key insight, and I would be remiss not to mention the sort of delivery side, because that's been the biggest bottleneck. I still think it's our greatest source of jeopardy. So we've been super lucky to work with Ben Deverman here at the Broad. I think he has sort of led the way, developing capsids that have a good shot. Like there's a strong rationale that they should be able to cross the blood-brain barrier. Um, we see examples out in the world, and again, the devil's in the details, direct injection into certain parts of the brain. I think can offer some benefit. We've seen a beautiful example in Huntington's disease recently in the context of diseases where there is a region of the brain that is most vulnerable, that's affected first, and you just need to get there and saturate it. Um, but Prion disease, I always have the most disappointing answer. People are like, where can we go to make an outsized difference? Where does the first prion form? And I'm like, it could be anywhere. So we're
Mm,
really like, I think our moment is now. It's now that we have the very first tools to start asking, what happens if we deliver a gene therapy to the whole brain? Can we get this done?
wow, that's really, thank you for that. You are very inspiring and obviously, um, I'd like to know a little bit more. So therefore, it's not enough that you figure out a way to edit the prion gene so that it's not working anymore. You have to get it not only to the right place, but in your case, sounds like you have to get it all over the brain, right?
Totally.
Okay. Um, and can I ask if this specific genetic change is something that is common to many people who have this prion disease? So that way, designing this therapy could potentially benefit many, if not all, people with this condition.
Yeah, that's a
Fantastic.
great question. Um, so I guess as background, people think of prion disease as very rare, and we sometimes hear one in a million. Um, I think that overstates the case a little bit. It's just that people die so rapidly that at any given time, there aren't a huge number of patients alive. But prion disease will actually kill about 1 in 6,000 people. Um,
Wow.
85% of those cases are sporadic, which means there's no known genetic or environmental trigger. It is probably a chance misfolding event that just takes off. But about 15% of cases are genetic. So there's my variant, but there's also a handful of others in the prion protein gene that make that event much more likely to occur at some point in adulthood. Um, Within genetic, there's a lot of different subtypes. There's like a whole alphabet soup. Sporadic disease is most often called CJD. Genetic, like my particular subtype, is called FFI. You hear all these different things, and I think of these names as a little bit accidents of history because you know
Sure.
what could we see 100 years ago? We could see what the brain looked like after someone died. And are there differences? Sure. But I think what do we have now that is our greatest asset? We have a single unifying genetic story about this one gene in this one protein. And what I like about the strategy of lowering prion protein is that it absolutely should be universal. And that's not even just true for all the human subtypes, genetic, sporadic, subsetting under that. But prion disease is like a disease of mammalia, and every example, be it in nature, in humans, wherever, every example of prion disease on Earth starts with PRP. So I
Mm,
think to me that's like precision medicine doing the opposite of subsetting. It's like precision medicine bringing everybody under one umbrella to say a
right.
single drug can be useful for all kinds of prion disease. I do think there's a meaningful distinction between genetic and sporadic just in terms of timing. When can we get to people? For sporadic prion disease, for the foreseeable future, we will just be racing to diagnose people as fast as we possibly can. And this is
Right.
something that I'm like constantly talking about. We have a great diagnostic assay called RT-Quick that we can run on CSF. I just need people to think of it and run it and get the answer. So that is like a battle that we'll keep fighting. Genetic, there's people like me who are walking around with this, 8.5 by 11 piece of paper that tells you your risk. And I think we would be remiss to not lean into that and treat people before this absolute lightning strike runaway train starts doing its damage. So I've come to realize that our mission is not just to develop the molecule, and it's not just to develop the supportive infrastructure that will allow us to test the molecule. It's also to kind of like try to crack open this nut of preventive treatment. I think that is what we need for brain disease, and that is widely recognized as being common sense. It's just a question of how to make our systems kind of bend to that mandate, which is pretty new. Being able to predict people's risk while they still look totally healthy, and maybe even their blood biomarkers, their imaging, all of that is normal. It's new, and it's a new challenge to try to like rise to. Um, but I think prion disease gives us a really, really clear example of outsized benefit, because if we wait till people are sick, they are very sick. So
Absolutely.
we have to keep working on that one.
Absolutely. So it sounds like you've raised issues of how to get the therapy to where it needs to go. But even like stepping away from the nuts and bolts of the treatment itself, it's like how do you identify the proper people who require that therapy? So that's even even once you even figure out if everything works right, it's like how do you get somebody identified so you can treat them in a timely fashion. Okay, so those are really some challenges. Can you maybe talk about some other challenges or limitations that come to your mind when we're considering like in vivo genome editing?
Sure. Um, I think a limitation in, and it's widely talked about, and it's not just our problem. Um, but I think as we are able to develop these medicines with great precision, and we're no longer kind of like throwing small molecules on cells and saying, these cells look better. Like maybe this will work. We don't know how. But I think the prior, not that drug development will ever be easy and not that success will ever be assured. But I think the prior for these genetically targeted medicines, at least being pointed in the right direction and having a shot at working in the clinic, is going up. And I think that's really exciting. And I think that like factors into risk benefit in an interesting way. Meanwhile, I think on the risk side, um, I, you know, challenge myself all the time, and I think my community too, to like lean into like our voice around what seems reasonable to us. Because a lot of our infrastructure for drug development is aimed at large common diseases where a lot of people will be exposed, and the benefits we're looking for are incremental improvements in health, not prevention or treatment of something that is universally fatal, right? And
Mhm.
so I think a lot about the challenges of just conveying risk benefit from the patient and family perspective. Um, there's there are going to be a lot of things that we can really only learn in humans. And I'm all for a thoughtful discussion about what safety testing can be done first and what we can learn from it. But I also want to challenge all of us to not lean on the sort of patterns and precedent of what safety testing usually looks like as a safety blanket. Because the truth is, lots and lots of things can only be learned in people. And that's scary. That's totally scary. But it's a Rubicon that we have to cross at some point if we think these therapies deserve to be tested. And I think we are looking at therapies that deserve to be tested. It is always going to be scary, and rightly so, to all of us that a gene editing therapy is very likely irreversible. And it's a one-time thing. And this is a huge pro and a huge con. Like the idea of getting an injection and then walking away from the idea of
Yeah.
therapy forever it's honestly surreal to me. It's like science fiction, and there's part of me that can't even like entertain that daydream. Um, I think that is like like um, just the prospect of that is fairly incredible. And there's actually sort of like functional and logistical underpinnings to that also. I think we know a lot of people in the rare disease world, including people on the lucky side of that story, for whom there is one or more approved therapies for their condition. It's a certain thing to sign up to fight for that therapy for the rest of your life. I would be very, very lucky to be in that position, and I'm here, I'm game, I'm ready to fight. But gosh, getting an injection and walking away is different. So I think that that is all really special. But yeah, absolutely. I think we bring a lot of caution to the idea that a change that to the idea of making a change that's permanent. And mostly, I think it just has to be all taken in context. I look at patients who are dying of symptomatic prion disease, and um, this, you know, when I tell my story, it sounds very dramatic and it sounds like a story that happened to someone else, right? But I know that it happened to me. And for all these people, right, they were just living some normal life that had nothing to do with prion disease. And then it just happened. And I think coping with the randomness of that is like a tremendous existential
Mm,
task. And um,
mm,
being told, as prion disease patients have always been told, there's nothing we can do. Like, we can't tell you why this happened, and we can't give you absolutely, like, we can't give you anything at all. That really does a number on people. It's like incredibly distressing to feel that not only is a terrible thing happening, but that terrible thing has no meaning, and you are being given no opportunity to make meaning out of it. And I think this is why people in our community show up in incredible numbers with incredible enthusiasm to do anything. And this includes natural history studies where there is no prospect of therapy at all. So I think
I
we
want
kind of need to
to,
honor people's
yeah.
desire to make something
Hmm.
out of this. And I think that we can trust them when they say, I know that this has very, very limited, probability of saving my life. Um, but here's the thing that I can offer. In this moment where everything is being taken away from me at like a
Yep.
rapid pace for no reason, here's the thing I can offer. I think that's very empowering for people. And I want to open up the conversation around letting people step into that.
Hmm. Hmm. Yeah. So for genome editing, really, it sounds like, um, you have one shot at it. You need to get it right. And I think sort of what I'm hearing is, and this is actually a conversation that we end up having with regulatory agencies is this risk, right? Because everyone knows that the risk of not doing anything is guaranteed. I feel like sometimes uh, the agencies come back to us and say, oh gosh, but isn't the risk of the delivery method, the risk of the vector, the risk of the editing, and the conversation is a little like, well, the risk of not there are harms that come from not intervening, right? So that's another conversation that again is not even directly like pertaining to the nuts and bolts of the editing itself.
Totally.
Um, so I'm looking through sort of the questions that I had in mind. I think you've actually touched on in some way or another like each and every one. I wanted to ask maybe one final question, which is maybe stepping away from the world of prion disease and looking at the world of like inherited neurologic disease or general inherited diseases. How do you see the potential for in vivo genome editing for uh, other conditions, other inherited conditions?
Yeah, absolutely. I mean, I think we're in such an exciting moment, and how to like marry the potential of all of this with like getting the details right for like each application is like the tension of our moment. I think so much will depend on, not not our ability to design the editor. I think we're getting good at that. So much will
Mhm.
depend on our ability to get the therapy where we need it to be, um,
Yep.
and to get to the patients at a meaningful time, and to move the whole process faster. This is like something that I like worry about. I think in the beginning of our quest, the jeopardy felt largely scientific. And that was a thing that we could like throw ourselves at. And, it was just me and Eric and our own four hands at the bench, and we could like work as fast as we could work. And it's not that there's no scientific jeopardy anymore, but I think really we've shifted into a paradigm of human systems jeopardy. And that's where I think, um, you know, the potential is enormous. And in some ways, we need to sort of jointly accelerate how quickly we can
Mhm.
move these things along. What about them groups together and is similar and is platform, and where do we share our learnings? What about them
Right.
is like a little bit bespoke, but still grouped on different dimensions, like the cells that you have to get to? And
Mhm. Mhm.
I think it's challenging for everyone, and there are no bad actors. I think like what a beautiful problem to have that our technology has moved so fast. Um, and I
Definitely.
don't see anybody in the whole ecosystem sort of standing in the way saying, we hope that this doesn't succeed. You know, it's
No,
all just a
absolutely.
question of like um, how creative we can be, right? Kind of together about like honoring the sort of totally new prospect for good that we have in our hands.
Thank you so much for sharing your story. It definitely is a tremendous inspiration, and appreciate your taking the time to talk with us today.
It was a total pleasure. Thank you so much.